Welcome to The Explainer. Today, we are diving into a truly fascinating journey to demystify the absolute core of our biological existence. We're going to explore a story that starts in, honestly, the most unlikely and kind of gross of places, with the gritty discovery of a mysterious substance in pus-soaked surgical bandages. And from there, we'll track how that bizarre starting point led all the way to today's multi-million-dollar medical miracles that are actively rewriting the code of life. We are talking, of course, about DNA.
All right, here is our roadmap for today. First up, the discovery relay race. Then, the anatomy of the blueprint. After that, we'll hit the central dogma, tackle the myth of junk DNA, and finally, wrap up with medical breakthroughs and CRISPR. Let's get right to it. Section 1. The DNA Discovery Relay Race. From obscurity to the double helix. You know, when most of us picture the discovery of DNA, we immediately imagine James Watson and Francis Crick triumphantly holding up that famous double helix model in 1953. But honestly, it was absolutely not a solo sprint. It was more like a 150-year relay race. It all kicked off way back in 1869 when a Swiss chemist named Friedrich Miescher was analyzing used surgical bandages from a local hospital. He found this bizarre, phosphorus-heavy substance he called nuclein. For decades, people thought it was just a weird chemical curiosity. Then, the baton gets passed. In 1928, Frederick Griffith discovers that harmless bacteria could somehow transform into deadly ones. By 1944, Oswald Avery and his team proved this transforming principle was actually DNA, completely unseating the long-held assumption that proteins carried our hereditary traits. Then, Erwin Chargaff figures out the chemical pairing rules in 1950. All of these brilliant scientists were running their leg of the race, teeing up the final puzzle pieces.
But here's a crucial twist to the story. We absolutely have to talk about the incredible and historically overlooked contribution of Rosalind Franklin. Her mastery of X-ray crystallography produced photograph 51. This wasn't just a neat picture. It was the undeniable, crucial visual evidence that DNA is shaped like a helix. Watson and Crick famously used her data, along with Chargaff's rules, to finally model the double helix. And while she was completely sidelined at the time, modern science recognizes that without Franklin's X-ray data acting as the master key, that puzzle simply couldn't have been solved when it was.
Section 2. Anatomy of the blueprint. Let's talk about actually building that double helix.
3 billion. I really want that number to sink in for a second. To grasp the jaw-dropping scale of the human genome, you have to realize that your 23 pairs of chromosomes, the ones you inherited from your mom and dad, contain 3 billion of these base pairs. If you were to take the microscopic DNA from just a single human cell, uncoil it and lay it out end to end, it would stretch almost 6 feet long. And somehow your cells managed to pack all of that neatly into a tiny microscopic vault called the nucleus.
So, how does this incredible length of DNA actually hold together? Well, it's essentially a twisted ladder, and the rungs of that ladder are made of 4 simple chemical letters: adenine, thymine, cytosine, and guanine, and they follow a very specific, perfect pairing rule discovered by our guy Chargaff. Adenine always locks together with thymine, and cytosine always locks with guanine, no exceptions. Because of this perfect anti-parallel pairing, if you know the sequence of one side of the ladder, boom, you instantly know the exact sequence of the other side. This beautifully simple pairing mechanism is the literal foundation for how life copies itself.
Section 3. The Central Dogma. From the code to the cellular factory.
Okay, so this elegant structure forms the basis of a core biological principle known as the central dogma. This is essentially the instruction manual for how that static DNA blueprint is actively used by your cellular factories. Think of it as a 3-step manufacturing pipeline. Step 1 is replication, where DNA unzips and copies itself so new cells have the exact same blueprint. Step 2 is transcription. This is where specific instructions from the DNA are transcribed into a highly portable messenger molecule called RNA right inside the nucleus. And finally, step 3, translation. That portable RNA messenger travels out to the cell's protein factories, the ribosomes, where its code is literally translated into functional proteins. And those proteins, they're the little machines that actually run your body, digest your food, and determine your physical traits.
Now you might be wondering, why does the cell even need RNA in the first place? Why not just use the DNA? Well, think of DNA as the original, ancient, highly classified master blueprint. It's double-stranded, contains thymine, and is simply way too precious to ever leave the secure vault of the nucleus. RNA, on the other hand, is like a disposable photocopy.
It's single-stranded, swaps out thymine for uracil, and acts as a rugged, portable messenger that can safely travel out into the cytoplasm to get the dirty work done. This brilliant system keeps the master DNA totally safe from damage.
Section 4. The Myth of Junk DNA. Time to unlock the genome's dark matter.
Get ready to unlearn a massive historical misconception. 2%. Out of our massive 3 billion-letter genome, only about 1 to 2% actually contains the genes that code for proteins. For decades, scientists looked at the remaining 98% and were just completely baffled. They literally labeled it junk DNA, assuming it was just useless evolutionary filler taking up space.
Well, spoiler alert, science has heavily corrected this myth. Thanks to massive research initiatives like the ENCODE project, we now know this 98% is far from junk. It's actually a highly complex regulatory control panel. This non-coding DNA acts as promoters and enhancers. Think of them as the dimmer switches that dictate exactly when, where, and how much of a specific gene turns on. It also forms telomeres, which are those protective caps on the ends of our chromosomes that stop our DNA from degrading over time. It even controls the physical compaction of the chromosome itself. So yeah, it's not junk at all. It is literally the vital operating system of your genome.
Section 5. Medical Breakthroughs and CRISPR, Hacking the Code of Life. Because after a century of uncovering these molecular mechanics, we're finally at the point where humans are learning to actively rewrite the instruction manual. And nothing illustrates this leap forward quite like Casgevy. This is a genuinely historic FDA-approved therapy utilizing CRISPR-Cas9 technology to treat sickle cell disease, which is a devastating genetic disorder that warps red blood cells. The way it works is pure science fiction made real. Using CRISPR, scientists take a patient's own stem cells and make a highly precise edit to a specific repressor gene called BCL11A. By basically cutting the brakes off this gene, the therapy reactivates the production of fetal hemoglobin, which is something our bodies usually stop making right after we're born. This healthy fetal hemoglobin floods the system, drowning out the sickled cells and effectively curing the disease at its genetic root. It is a total game changer. But to truly understand the profound impact of this tech, we really have to look past the biochemistry for a second. Listen to this incredible quote from Victoria Gray. She's a mother of 4 and was the very first sickle cell patient ever treated with Casgevy. Before this therapy, her life was defined by constant hospitalizations and excruciating pain crises. After the edit, her life was completely transformed. She said, "I feel cured. My life has changed dramatically with just a leap of faith." We have officially transitioned from talking about academic, microscopic mechanics to profound, life-saving human impact. However, and there's always a however, this incredible leap forward brings us face-to-face with a staggering number. $1,930,000. That is the estimated cost per patient to make this new CRISPR therapy viable. While the science is undeniably miraculous, this price tag highlights the intense economic and accessibility challenges facing modern genomic medicine. Figuring out how to actually deliver these complex, bespoke cellular edits to the millions of people worldwide who desperately need them, that is the next immense hurdle for us to clear. So, looking back, we have traveled an unimaginable distance, right? From Friedrich Miescher analyzing pus-soaked bandages in a hospital back in 1869 all the way to an era where we can deploy a nearly $2 million microscopic scissor to cure genetic diseases by directly editing the fundamental code of a human being. The relay race definitely continues, only now we aren't just trying to read the genome, we're actively rewriting it, which leaves us with a pretty profound question to ponder. What part of our blueprint will we rewrite next? Thank you so much for joining me on this incredible journey through the explainer today. Keep questioning, keep learning, and I'll catch you next time.